a very quick introduction, 5-10 minutes maximum, about the Bond-in-a-Box platform in general, and then we'll move on to presenting more of the user interface itself and case studies, and also the details of how to work with Bond-in-a-Box. And in the end, you'll have time to work with ourselves being there for support live. There will be breaks in between. We won't do 3 hours straight. Um, and that's about it. The meeting is being recorded, and as well, that's also for all the theoretic part. But we'll stop the recording for the part where people work. Actually, I hear echo of my microphone. Actually, maybe it's from Jory.
It is.
Okay, it's a bit confusing. Thank you. Okay, so all right. So GEO BON, as you probably know, is a large number of members which you are most probably part of or interested in. We're known for EBVs, ecosystem service variables as well, and essential ocean variables that are let's say, specific EBV. The bonds can be really global, like based on a thematic like marine, soil, freshwater, but then it can also be regional for a specific country or region. Where it's interesting in Bond-in-a-Box is the way we'll be able to share analysis pipelines. So your workflows can be shared within these networks. For example, marine things that work in one place in the world might work elsewhere. So making it available more easily can benefit your community of practice. And whatever works in the national bond can be transferred to another national bond just the same way. Bond-in-a-Box is many things, and people get confused about it. There is a tool catalog that is what it started out with, and we have Christian on the call, who was the developer of this initial website. So raise my hat to Christian. A catalog of tools that are useful for biodiversity observation networks. And later on, we added also a catalog of projects in order to put the projects online where they were, let's say, less visible, sometimes hidden in spreadsheets or in local communities that we don't know about. And in Humboldt, they had spreadsheets full of this. So what they did is they created this. Instead of making a portal for Humboldt only, they made the GEO BON Bond-in-a-Box portal for projects, so anyone can use this to gain visibility for projects that don't have a website, and that are not necessarily easy to find. But there was also another big need that we saw. GEO BON's vision is to help, because we can't do that alone, help establishing a global biodiversity observation system. It's analogous to the GCOS, for those who are familiar with the climate ecosystem. And in order for all these networks to communicate together, well, it's something's already a bit happening. So organizations are striving to put the data the most fair possible, to have interoperability between organizations. So it's sort of happening whether we give it a name or not, and whether we formally organize it or not. But if we zoom into the analytics section here, we already have good ways of sharing data. It's continuously improving, but good ways of sharing analysis workflows within the community is usually packages and papers and dedicated web portals, which goes a certain way, but there's a need for a platform that allows to share these things visually, easily for decision makers, or for not necessarily decision makers themselves, but the scientists working with decision makers, to pick on these analysis much more easily than starting from the package and having to code everything. So we introduced the Bond-in-a-Box modeling tool, going from this knowledge to action perspective, trying to integrate the data and the models. We're not hosting the data, we're more hosting the models, but the models do transform the data into useful insights. So we calculate these EBVs and indicators based on a curated open source repository of community contributed analysis pipelines. So it's really the open source philosophy, and this is something that's powering the Bon in a Box philosophy. Um, we're striving for people to share and work together, and it's also a bottom-up approach. So a bit like in this very workshop, those who are coming to us with what they want to share, and then we help them along the way so they can achieve their intentions, and that it can be can reach a wider public or be shared in their community of practice, for example. I'll just put that in image a bit with this this figure by Michael Katchen that is in the preprint. If you might have read our preprint, you can see the workflow from biodiversity observation networks that collect data on the top, and then merge this with the raw data from the satellite as the in situ data as well into a pipeline. So the pipelines are on the right there. They go from one script to another, the outputs of one script being the inputs of the other script, all the way to the EBV, to the indicator, and then goes back to informing the monitoring and conservation efforts, and maybe even reporting to the CBD. This happens in this diagram with Colombia. But what if, well, they did actually. Colombia formalized some of their workflows into Bon in a Box pipelines through Humboldt Institute. And when they do this, what they allow is that we can change the country there for another country much more easily than if the other country or other region had to start from scratch and recode everything. So it allows the same wheel of informing the monitoring and conservation effort from their data to happen much more easily in other places. So just to crystallize the idea, what is it for? Computing policy relevant biodiversity information, EBVs indicators, running any analysis that is relevant to biodiversity monitoring at any scale. What I mean by this, we speak of the CBD, but it's not just for the CBD reporting. It can be in communities as well. It could be city scale, park scale. Actually, the way we build the framework, it can calculate anything that can be coded in R, Julia, and Python. But we, of course, in the context of Bon in a Box, we specialize on biodiversity. And then prioritizing areas for monitoring. This is a research in progress, but a very useful one is to be able, based on the uncertainty and the results of number 1 and 2 there, calculate where we should go to have a better idea of number 1 and 2 and have more sampling to get better results. Then solving all these challenges, biodiversity monitoring is just the tip of the iceberg. You know, there are data problems, collaboration is not always easy, resource. When we speak of resources, we often think time and money. But then when, sorry, I got distracted by the chat, but it's also resources of knowledge. If the scientists that know how to do a certain operation are just in one place in the world, in one lab, then it's a huge resource constraint. Well, when everybody in the world wants to calculate the same thing. Also, the code is complex. When it exists, sometimes indicators are conceptualized and not coded. And when it exists, is it properly documented? Is it easy to pull up together and run? That's another question. And the data, I'll just come back on the data quickly. We are not data providers, though we have a stack catalog because it's convenient. Our goal is to feed on the data from many sources where the data is best hosted. And all of these problems cannot be solved by a single platform anyways. Let's see how we tackle collaboration. So yes, we are open, transparent, and modular. Transparency being that you can see everything that happens in the process, all the intermediate results. Open is the open source, as I mentioned. The modular is that what is done in one pipeline can be reused in another context. You'll see that with Jory in a few moments. And then operationalizing existing knowledge, because we don't want this really good science hidden behind paywalls and papers and sometimes hard to reuse. We want it to be as easy to use as possible. And the fairness of the analysis process is something we're starting to hear, because we speak a lot of fair data. But then, is the analysis of the data fair? Can we find the analysis that was used? If it's in a repository somewhere, yes. Is it accessible? Can I easily pick it up and use it? Is it interoperable with other data sources than the one it was conceived for? Is it reusable in other contexts, or even to recalculate the same thing? So the fairness of the analysis process is something we're striving for with Bon in a Box. And also increasing scientific rigor, because, well, you have to go through an additional round of documentation in order to put this on Bon in a Box, but as well, there is some peer review that will be going on. I'll just show how Bon in a Box can integrate tools from different communities. This is a slide we did for the Biospace 25 conference. So we have the EO community that have their own set of tools and work more in Python, and the biodiversity community that has their own set of tools and work rather in R. Bon in a Box runs R, Python, and Julia for the coding languages, and, well, we could eventually add others. And connects to all these platforms simply through the scripts that people write. And it allows to merge these two sets of tools in a coherent way, because the pipeline engine just seamlessly passes from one to another. So we're able to get the data, these generic data, and make an indicator out of it. But if you go to someone in a small region with something you calculated with global data, they're probably going to laugh at your map or at your results, because there's something missing, local data. So we also encourage the pipeline designers to always leave the door open for the complementarity of local and global data. You can write a pipeline that only works with global data, then someone will have to go in and modify it in order to put his own local data. Or it can be designed to accept both and merge it from the start. But the pipelines can always be modified. So if you have, let's say, a very, very specific format of local data, you could always put in a script that does what you need in order to import this data into Bon in a Box. So we have all, these are the first pipelines we officially have. More coming, especially with this workshop. I'm eager to see what will happen. So we had capped the registrations at 40 participants, and we have 40 participants. So I'm really eager to see what will come out of this workshop. And what you see there was contributed by people from Switzerland, Canada, and Colombia. Now I'm just going to go through a little question break. I've sent a lot of general information that many of you maybe already knew, but for some it might be new. So just let us know if there are any questions. No? Okay, so we'll forge ahead with the tour of the user interface. I'll just stop sharing for a second. We can hear a dog. So now we have the mics working. And I know it's not in Jory's office because she's right there behind that red door. Okay. Can you see the Bon in a Box form now? Thumbs up, someone? Yes. Okay, good. All right. So when you get to this Bon in a Box user interface as a user. So I'm going to focus on the user first and then on the contributor side. As a user, you probably want to run a pipeline, which is on that tab. You can go and search here for whatever you want. So for example, you can go and search for a specific pipeline here. But I'm just going to use this other one I had because it already had some data there. Oops, I just lost it. Okay. This is a species distribution modeling pipeline. You can see the authors. You can see a quick description. Could be a bit more lengthy. External references. You can also reference papers and put a license on this. And then the form. So once you have selected your pipeline and read the metadata, you'll have to input the parameterization of your run. We call this a run in our jargon. So you fill a form, you run it, it's called a run. For each input, you have a description that was chosen by the pipeline contributor. You just go through the whole form. So it's usually values or file paths, line cover classes, things like this. And then you click run on the pipeline. Then the results will appear after the pipeline has computed. This can take like 20 minutes, for example, for this SDM. And you can see the top section here is the outputs that were selected by the contributor as being the most relevant ones. It's not necessarily all of the outputs. It's the one that you want to show in priority. But for the transparency purpose, when someone is doing the analysis, you might want to know what is happening in every single step of the pipeline. And so you might want to go here. The logs are there. You can see the selected raster after a step that removes the collinearity in the predictors. So you can see all the results individually. This is for the transparency part of it. And this will display tables and TIFFs and data types like this. For those data types that are not supported, there is always the possibility to click on the download link and visualize them outside of Bon in a Box. It works sort of with any possible file type because in the end, you can always download it and visualize it with another tool. There is this button here, see in results viewer. That is what you will want to do when you present the results to someone else. Probably you don't need all the intermediate results. You don't need the logs. So it's a simplified user dashboard where you can see only the outputs that were selected. This will be the red boxes a bit later on in Jory's presentation. The outputs that were selected by the contributor. For example, here we see the prediction of this SDM and the other things that were considered important, such as the presences of the species on the map and uncertainty, or maybe some of the predictors here. You can always access with the run history to older runs that you did on this instance. So I just mentioned the word instance. Bon in a Box is something you can run on your computer, as you probably know, because part of the requirements for this webinar was to install it locally, but it also runs on a server. So for example, here we have our results server, which is online, and this is the one we use to show results on the website. And we also have instances, larger instances, in order to provide computing capability. If you need 180 gigabytes of RAM for a specific analysis, we can lend you that instance. Then I'll go on the contributor side. For this, I'll move back to the presentation. Okay. So Jory will show you how to use it, but I'm going to point to you the different sections of the UI. This on the left is the list of available steps. A step can be a script, or it can be a full pipeline that you can drag and drop in the main pipeline editor canvas. In the pipeline editor canvas, this is where you'll assemble the script, connect things, put the outputs, etc. You can see here, a step, for example, is a box. The explicit user input is the green box there that we have connected to many other places, because if there's a start date here and a start date here, well, you don't want start date to be there 2 times in the form of that the user will fill. Leaving an input blank just automatically puts it in the form for the user to run the pipeline. So we can call this an implicit user input. On the top right part, you have the file commands. The layout button will just put the boxes using an algorithm to space them a bit more than how we squeezed it here to fit the screen. But and if your pipeline becomes messy, you might want to save it first, because we don't have an undo yet. Save it first, and then try the layout command to see how it works. Usually it works pretty well. Then you can load either from a file on your computer, the pipelines are saved in JSON, or from the server, which is your local instance. If you are on your computer, that is, well, also on your computer, but specifically in the Bon in a Box folder tree. And then you can clear, which is start again from new, save, and save as. On the right side here, you see there's a pane here saying 9 inputs, 2 outputs. So this pane can unfold, and here you can see and also edit the details of the inputs and the outputs. And the pane just below is the metadata one that you will have to fill in order to be able to share your pipeline with people, because people want to see your metadata, want to know what your pipeline is doing, want to know your references, etc. I'll leave you on the technology stack slide. If there are any questions, we have another moment. Otherwise, we'll go for Jory's demo. I think you're all eager to start coding. All right. Go ahead.
Hi, everyone. I'm Jory. I'm the biodiversity data scientist working for GEO BON on Bon in a Box. So I'm helping contributors integrate pipelines into the tool. I think we kind of jumped into it and didn't do introductions. So that's me.
We had
and
so many
Jean-Michel,
technical problems.
Jean-Michel, if you could just go back to the agenda slide. I'm not sure if you went over that, just because I want everyone to be somewhat aware of how this is going to go. So Jean-Michel, I'll introduce him. He is the developer of the Bon in a Box project. So all technical issues go to him, and all R or Python coding issues come to me. So this is just the agenda of what we're going to do today. We already did the introduction and the tour of the user interface. I'm going to talk a little about conceptually how to plan a pipeline from a workflow that's already existing. And then we have a couple lovely volunteers, Emily and Eric, who will talk about some pipelines that they're integrating into the tool and how they sort of conceptually broke that down into a pipeline that they're going to put in Bon in a Box. We have a short break after that. Then I'm going to get into some more technical details of how to actually integrate scripts into Bon in a Box into a pipeline. We're going to talk about a few documentation standards, and then we'll have hopefully about an hour to do some like breakout groups, solo work, and then if you guys have any questions for us while you're doing this, then we will be available to answer those. Okay, you can go back to the slide. Okay, Great. So I'm just going to break down a few concepts before we dive into the examples. So the first one is a script. So that's just an individual step in a pipeline. You saw the pipeline editor canvas before, and that's just a single box. So it's a script in R, Python, or Julia. It has inputs and outputs in specific data formats, and then all of the scripts that are available go into this script depot. So the idea is that it's fully modular. All of these scripts can be reused in other pipelines, and it just reduces the amount of work that other people have to do on coding. I can change the slide. So each script is accompanied by a description file, and this is just a YAML file, which describes what the script does, the author, the license, etc., and then specifies the inputs and outputs of the script. So it describes the inputs and outputs, names them, and then has examples, and then has like the file format of the inputs and outputs. This YAML file then connects to that user form that you saw earlier in Bon in a Box. So each of the inputs and each of the inputs is generated in this form, and then the user can just fill it in, press run, and then the pipeline or the script will run in Bon in a Box. And then the pipeline is just an interconnected set of scripts to run an entire workflow. So those inputs and outputs allow you to connect scripts. So you can connect like a raster output of one script to an input of another. So instead of having to run all of these things sequentially and manually, Bon in a Box can just run that entire workflow as a pipeline. Are there any questions on this? All right, we'll keep going. Stay on the... Jean-Michel, can you stay on the presentation? And change the slide. Okay, so when you're sort of thinking about how to conceptually break down your workflow into a pipeline, this is the kind of things that you should be thinking about. So you have this workflow, it might be in one script, it might be in several scripts, but however it's broken down, it's important to think about how this can be sort of modularized. So we want each step in the pipeline or each individual script to sort of do one task and output an intermediate result. So this is sort of takes more time than you would think it would, but you really want to think about how your workflow can be broken down into these modularized steps, and think about maybe some steps that could be reused in other pipelines. So the idea is that in this workshop and in the next 2 weeks, you will think about how to break down your workflow into these steps. Think about the inputs that you want and what file formats you want them to be in. Think about the outputs and what file formats those should be in, and then how those are going to connect to your subsequent steps. And then what you want the pipeline output to be. So what you want the person who's running the pipeline to see at the end. Are there any questions about this? All right, we've got, if you update an input. Good question. So yes, when you are running, well, it's a bit more complicated with inputs and outputs because you have to reload the pipeline, which we're working on making that easier. But if you like changed a parameter downstream, then it caches the previous results. So you don't, if you rerun it, it won't like rerun the full analysis if you didn't change things like upstream, if that makes sense. But currently we don't have any option to like stop the pipeline and then continue running it, but that's in the near future.
Well, we have a stop button, but there's no automatic, like someone can crash the pipeline if a result is invalid. So it's something we'll see, but you can always stop manually and restart it manually. And about the caching mechanism, so if you run a script, make a change in the R code and run it again, it's gonna rerun it. It will detect that the file was modified.
Yes, but if you changed R code like downstream, then it wouldn't run the rerun the previous steps
Exactly.
if you haven't changed those, or if you haven't changed the parameters. If that makes sense.
Yeah, exactly.
Okay, I think you can change the slide. So this is just an example of what this sort of diagram could look like. It's a bit messy, sorry. But it's just an example of like a species distribution model. So you have a script that gets the data from GBIF or OBIS. You have a step that pulls environmental layers from a stat catalog. You have the file outputs that you want. Some of these you might also want to do as some of these intermediate results you also might want to have as like final pipeline outputs, like the environmental layers. And then, yeah, so the SDM model, cleaning data, etc. So you're creating this sort of modular workflow. So then a lot of these, like getting, GBIF data or pulling environmental layers or cleaning GBIF data can then be used by other people so they don't have to rewrite that code. You can change the slide. So this is just sort of an overview of the contribution workflow. You take an existing workflow, make it modular, and also as generalizable as possible so that it can be run in other areas at different scales for different species, etc., so that other people can actually use it in their context. Build the pipeline, document it. We'll talk about all of this later. We're implementing a peer review process that will be relatively similar to a manuscript review process, a little bit less formal, but the pipeline will go through peer review from GEO BON experts and then published in BON in a Box and then hopefully used by other people. You can change the slide. Okay, so now we have a couple lovely volunteers who have volunteered to talk about pipelines that they're putting in BON in a Box and how they conceptually broke these down. So first we have Emily Beasley from University of Montreal talking about a rabies model in Ontario. So Emily, take it away.
Yeah. Hey, I'm Emily. For the purpose of this short little presentation, I collaborate with a lot of governmental agencies like the Public Health Agency of Canada and the United States Department of Agriculture, while they still exist, on managing zoonotic diseases such as rabies. And what a lot of these managing agencies are interested in is basically a low coding way to make like rough predictions of rabies dynamics. Things like if they change their vaccination strategy and vaccine placement, how could that change rabies dynamics? They want to look at movement corridors of the hosts of these viruses and potential effects of anthropogenic translocations of the hosts. And they want to use this to basically generate a quick, easy prediction so they can either collect more data as needed or send it over to their internal statistical folks and do a little more in-depth analysis. So you can go ahead and change the slide. So here's kind of what the agencies wanted. They wanted to be able to take data that they already have in formats that they're used to working with, such as land cover data shown by A, case data labeled with a B, and shape files of their vaccination strategies. Put those in an agent-based model called the Ontario Rabies Model, and then yield things like heat maps and epidemic curves and stuff like that. So changing this workflow into a pipeline, you can go ahead and change the slide. Basically everything here on the left, so the makegrid.py or gridattributes.r, that's all data cleaning. The sampy.py file, that's the main agent-based model. And that's going to spit out outputs as a CSV, which is going to go to an R script that makes some easy to read figures. So yeah, that's the general idea.
This is awesome. Thank you, Emily. Does anyone have questions? All right. Oh, Lena, hopefully you can turn on your mic. Don't think we can. Okay. I don't think we can hear you. Or at least I can't.
Yeah, we can't hear you, Lena. It looked like it was on mute.
Okay. Probably easier. Yes. So
yeah, Python and R, same pipeline. Yep, that's correct.
And Bon in a Box supports that because it's just outputting like file formats that can be input in any script. So like a GeoJSON or a CSV. So that allows you to connect different scripts in different coding languages. And right now we support our Julia and Python and Bash.
And Bash if you really want to.
So that's another cool thing about Bon in a Box. All right, so now we'll move on to our second volunteer from ABMI, and yeah, so Eric made or conceptualized and also has started putting in Bon in a Box a backfill model for natural vegetation. So Eric, hopefully you can turn on your mic. I know you're having issues with that earlier. But yeah, go for it.
Hi, everyone. So I'm Eric, GS specialist working at the ABMI. And it's a work, so can we go to the next slide, please? So I will introduce Brandon Allen, who is our senior ecologist at the ABMI, and he will do the first part of this presentation to give you some context about ABMI and what the ABMI Science Center is doing. And then I will present the workflow.
Awesome. Thank you, Eric. Yeah, you can go to the next slide, Rory. So yeah, just because we didn't know who was on this call, so the ABMI is the Alberta Biodiversity Monitoring Institute. We're an independent nonprofit that has been operating in Alberta since around 2007, and we do a lot of work tracking changes in wildlife and their habitats across the province. So we do a lot of reporting, developing indicators, and work collaboratively with folks in across all industries, so forestry, oil and gas, government, and NGOs. Next slide, Jory. So the indicator that we work on a lot is the Biodiversity Intact Index. I'm sure a lot of folks on this call are very familiar with that index or variants of it. But there are a few different ways that people seem to calculate it. So you often need a reference condition, and sometimes folks use protected areas, so just nearby local areas, to determine baseline reference conditions. Sometimes it's through landscape simulations, looking at things like landscape as a pre-colonization. And at the ABMI, we use what we call a backfilled vegetation or a defootprinted vegetation layer. So Eric's going to walk through the pipeline about how we use current vegetation information and backfill human footprint features to get an understanding of what the landscape would look like if footprint hadn't occurred in an area of interest.
Okay, so the ABMI came up a long time ago with this workflow to defootprint or backfill, and it's a polygon-based vegetation inventory. Most of the vegetation inventory in Alberta are polygon-based. They are the results of photo interpretations. So how do we defootprint? This is the workflow. we have human footprint. We want to backfill, so we split them first in non-linear human footprint and linear human footprint. Linear human footprint are mostly roads that would be diced into small polygons and merge with neighboring polygons. So now for the non-linear disturbances, we split them in two categories because this is a different set of rules for the backfilling. We have harvest area for timber. So we need to backfill this area with treed vegetation. And we also have a criteria on the edge of the vegetation, treed vegetation. We should have all trees 80 years old at least. So we will develop tools for, we could develop tool for GEO BON, one tool to backfill harvest area, one tool to backfill other non-linear disturbances, and one tool to backfill linear disturbances. And then another tool to merge all this backfill human footprint and the native vegetation around them to create a reference layer. I won't go more into details. And if you have questions, I can answer them later. Next slide. So for this workshop, I started working on the first script to integrate into BON in the box for the backfilling of harvest area. So this script requires as input native vegetation suitable for harvest area, for backfilling harvest area. So it means that we will need to know tree species and also the origin year of the tree, because we will need to compare the origin year of the tree with the harvest year that the other layer, the human footprint harvest area polygon. Sorry. We have two inputs, native vegetation with tree origin, and you will need also the harvest area for timber polygons that needs to contain also the harvest year. And we will be comparing harvest year and tree origin to determine if the tree is old enough to backfill the harvest area. So this is the tool I've been working on. How it works, next slide. So it's we look first at if there is any suitable vegetation in a 50-meter buffer around your harvest area for the backfilling, and we select the dominant one if there is 2 or 3. And we assign the vegetation type and the age of the tree to the harvest area. If there is no suitable vegetation in 50-meter buffer, we look at the closest in a 2-kilometer buffer. And if there is none, we look in a 5-kilometer buffer. So the difference between 2 and 5 kilometer, in the 2-kilometer buffer, we still assign the edge, the tree patch used to backfill the harvest area. We don't do that in the 5-kilometer buffer. Then if there is no suitable vegetation in the 5-kilometer buffer, we need a backup layer. You have to create a backup layer, and we have a few cases where we need these backup layers to assign the vegetation. So how do you create this backup layer? Depends on the other data you have available in your region. At EBMI, we are combining several layers like vegetation derived from soil type or vegetation, dominant vegetation in a natural subregion. And this is our backup layer because we don't have information coming from the main source, the main vegetation inventory. Next slide. So I want the step for, so I already scripted my and integrated in BON in the box. The script, it's a work in development, and I hope from this workshop I will be able to tailor it to make it really fit with the philosophy of a BON in the box. I will maybe remove some inputs and add some inputs like maybe projections and things like that, some consideration that make it useful for other region and different scales. So it was coded in Python using pandas and GeoPanda, open libraries, and the data storage format is a geo package. So it was quite a bit of work because all my scripts are written using ArcPy and S3 ArcGIS Pro. So I had to do some translation, but it worked, and yeah, it's a working script right now. Thank you. Any questions?
Thank you, Eric. I have one. Because you have this 52 kilometers, 5 kilometer or vegetation. Can you use this to output some sort of uncertainty map based on how far you had to fetch your information?
We track the information. So in the layer, I had a field backfill method, and then okay, the question is, when I do this backfilling, I track the origin of the vegetation. So you have the field telling you it's come from the 50-meter buffer, it's come from the 2-kilometer and 5-kilometer. So later on, you can look at it and map this information and get a sense of certainty you have in the result.
Interesting. Thanks.
All right, we're going to take a short break before I dive into some nitty-gritty of how to actually adapt a script to put in a pipeline in BonInABox. I want this to be very interactive, so if you have any confusion or questions while I'm doing this demo, then please don't hesitate to ask, and hopefully your microphone works.
I actually have a general question.
Sure.
I don't
Go for
know
it.
if you can hear me. Okay. I was wondering if there are any requirements to join as a pipeline. Can it be any biodiversity indicator, or should it be an essential indicator, or?
Yeah, so I think we briefly mentioned this in the presentation at the beginning, but we like any indicator that's in the GBF or outside that you think would be useful for other people to use, or just any like workflow. Like we have species distribution models in there, which is not an indicator, but it is an EBV, I guess. But pretty much anything that you think would be useful to share and that you think other people would want to use. So it's not strict.
Okay.
Requirements.
It
Yeah,
must
as
be
long as it's like this.
generalizable.
Yes.
Go ahead.
So it has, other people have to be able to use it in different contexts, and then also it has to be like a scientifically verified method in some way. But that's what the peer review process is for as well. So.
Okay, thanks.
All right, so I'm gonna dive into it. So all or most of you should have installed the platform before this workshop. So that includes cloning the GitHub repository. So you should have a folder somewhere on your computer that's called BonInABox pipelines. So right now I have Visual Studio Code open to my BonInABox pipelines folder. You don't have to do this in VS Code. You can do it in RStudio or whatever other code editing software you want to use, but I will be using VS Code for this example. So right now I have the BonInABox pipelines folder open on my computer. And when you're creating a new pipeline, you want to create it in this scripts folder. Oh yeah, and before we start, we work using GitHub branches. So probably before you start working on your pipeline, you want to create a branch for your pipeline and name it. So I have created this folder in the scripts folder. So you want to create a folder that essentially names your pipeline and describes your pipeline. So we have like biodiversity and Taxonomy Index, data, which is a really like sort of general getting data folder, but you don't have to put anything in that. And we have all of the individual pipelines here. So I created this one called workshop example. And for my example, I'm going to use a script that I coded, a pipeline that I coded for the Biospace conference, which essentially uses the program OpenEO, which is an API that calls EO backends to retrieve satellite data or processed layers. And then I do some stuff with it. So I'm doing this because it uses both a Python script and an R script, so I can show you guys how to use that. But for this, you do need an API client ID, so you can't run it right now, unfortunately, unless you get that, but it's pretty easy to get. So anyway, so the first step is that I'm going to create a file within my folder. Sorry, one sec. Okay, so this pipeline, I'm taking the Copernicus data ecosystem phenology layer. So I'm going to call this summarize phenology.python. And then every time I create a script, I also need to create a YAML file that has the same name. So I'm going to create a file called summarize phenology.yml. So now I have these two files, my Python script and then my YAML file that goes along with the Python script. So the first thing I want to do is import the packages that I need. So for these ones, I'm going to import in the usual way, just all of the packages that I need. One thing to note is that every time you run a script in Bon in a Box, you have to import JSON. So whether you're using an R script or which you would import RJSON or a Python script, you have to import that to run it. And then my second step is that I'm going to create a YAML file that describes my script and has the author. I'll paste this for you so you can see. So this is how you're going to start every single YAML script. You have, sorry, I missed one here, but the first thing should be script and then the name of the script that you're describing. So summarize phenology.python. So now it knows that it's describing this Python script. The name of the script, which is what's going to appear in the UI of Bon in a Box, the authors and all their information. In this case, it's me. The license that you want it to have, I'm doing CC BY here. And then a description of what the script does so that if someone wants to use the script in another pipeline, they can click on it and see what it does. So this one summarizes yearly phenology data. For a country, it's Europe only because that's what the layer contains. It uses OpenEO to pull the plant phenology index. So then my next step is that in my script, I'm going to load my inputs through Bon in a Box. And we have this nice function to do that called BIAB inputs. So I'm going to call that data equals BIAB inputs. And I'm going to write some code. So this chunk of code here essentially connects to OpenEO through an API. And I have my client ID and client secret, which in Bon in a Box will be contained in your environment file. So it'll be called, when you're setting up Bon in a Box, you had to change this to runner.env, and that's where you put all of your API information for GBIF, for OpenEO, for any place that you want to get data with an API. And so my next chunk of code is going to be essentially creating this data cube where I'm pulling data from OpenEO with these different parameters. So I have the spatial extent, the layer, the temporal extent, and the bands of interest. So let's say I've written this code for my area, but now I want to input this into an actual Bon-in-a-Box pipeline. So I'm going to change some of these parameters to be able to interface with Bon-in-a-Box so that the user can input these. I will copy and paste this to save you the painfulness of me having to type it. So this is what it's going to look like. So instead of putting in coordinates for my bounding box, I'm going to put in an input that's put in by the user. And you can see this hasn't been defined yet, but we'll do that in a second. So I'm naming these inputs right now. So I've named this one B box. I've named, for my temporal extent, I've named it start year and end year. And I've named the bands. So pretty easy to understand. I'm going to go ahead and delete this. So now I actually have to define these inputs. So I'm going to go back to the top of my page. After I input the data here, so this is what it's going to look like in Python. So I named it bounding box down here, bounding box, start year, end year, bands, and season. So now I've defined these inputs for creating this data cube and pulling this, these layers. So now I have to put these inputs and define them in my YAML file. So I'm going to go back to my YAML file here. I'll save this because it's good practice to save things. And I'm going to write inputs. And then I'm going to define my inputs here. I just pasted a lot of stuff, but essentially each input, you're naming it here. So the same name that you put here, you want to put as the name of the input. The label, so this isn't a great label, so this is more a human readable label, so I'm going to change this to bounding box. Description, so this is where you have the description of the input, so the user can know what they should put and what format it should be in. So here it's xmin, ymin, xmax, ymax. The type, so that's the type of data it's going to be. So this is a float array. And then an example. So you always want to have an example, except that I know I'm going to connect another script for the bounding box, so I'm just going to leave that null. But even if the example is null, you still want to write example. And so this is my start year, very similar. Start date for the phonology. This is text, because I'm putting it in quotes, but it could also be integer. I have the season of interest, so which growing season, and then this is long, but the band of interest. So this is all of the bands that are contained in the data set, and I'm allowing the user to choose what band they want to pull. So this is an options array. So I'm defining predefined options here, and the user can choose one or more of those. You can also just have options without an array, where the user can only choose one option. Does anyone have questions about this so far? All right. Michelle, I'm going to let you man the chat.
Yeah, well, for anything that's in the chat, me and Guillaume will handle it on the side. But if you want to raise your hand and ask a question to join, it's fine.
Oops. Okay. Yes, Ben?
Yeah, well, I think he's... Sorry, there's a bit of an echo. Can you still hear me?
Yes.
Shoot. I had to switch between devices. I put it in the chat, sorry.
Okay. All right, I'm going to continue then. So now I'm going to show you how to do a slightly different thing. I also want the user to be able to choose the spatial resolution. So I'm going to go ahead and define that input up here and call it spatial underscore resolution. But I also want the user to be able to choose whether they want to resample it to a different spatial resolution, or if they want to just leave it at the spatial resolution that the data is already in, which I believe is 10 by 10 meters. So that's pretty fine resolution. And so I'm going similar to writing normal code. I'm just going to do an if-else statement. If spatial resolution is none, you can just leave it as it was, or else you can resample it to the spatial resolution of choice. So that allows the user to leave it as blank if they don't want to, change it. And I'm doing the same thing for polygon here. If they don't want to crop by a polygon, they don't have to, but if they do want to, they can. And I'll add polygon as a input. And then I'm of course going to add these as inputs in my YAML file. Which I think I already did. Okay, perfect. So now I'll quickly show you how to write an output, and then we can move on to how to do this in an R script.
Jory, maybe I have a question.
Yeah, sure.
So all these data, the box inputs in your YAML file, I just didn't understand where they're coming from. So if you're going back to your YAML file, so
Yes.
all these data, like for instance, like the options, and so is this just predefined by you, or is it coming from someone from somewhere? So I don't get it. From where do you get this?
Yeah. So right now, all of these here, I'll just quickly show you. So right now, all of these are just defined by me. So if I go to single script run, summarize phonology, you can see all the inputs here. So bounding box, polygon, date, and etc. So this is just for the script. But as you'll see eventually, I don't want someone to have to input a bounding box. So I'm going to take another script that pulls a bounding box and a study area polygon from a database so that they can just input the name of a country instead. So in that case, bounding box wouldn't appear here because I've connected it through inputs and outputs. But I'll show you more on that later. But does that make sense?
Yes. Thank you.
Yeah. All right. I realized I accidentally pasted this twice. I'm just gonna fix that. Okay. So now I'm going to write some code to actually pull this data and fetch the rasters, etc. That's not so important to look at. But now I want to actually output this in Bon in a box so that it appears as an output. And so I'm going to use this function BIAB_output with the name of the output, which I'll soon put in the YAML file, and then the name of it in the script. So I have these raster output list that I've created. And so this is the name of my actual output, and this is the name of the object in the script. So now I'm going to also put this in my YAML file. Outputs. I'm going to call it rasters, because that's where I called it here. Have the label, so it's the rasters of phenology that I pulled from OpenEO. A description, and then the type. So in this instance, it's a GeoTIFF array. So it's multiple raster layers that are being output separately. You don't have to have an example for the outputs, but you can if you want to. So now just quickly, I'm gonna also write some code for summarizing this data into a CSV time series. I'm just gonna paste this quickly. It's not so important, but I'm also outputting this as a time series, and then in my outputs, I would put that, I'm going to call it zonal statistics because it's the average over my polygon of interest. And then it's a text/CSV. So the last thing you want to do to put this script in Bon in a box is to specify a conda environment. So I'm going to look at the packages that I loaded here. And then I'm going to look at in this runners tab, there's a Python environment YML, which has all of the dependencies that are already loaded. So if you're only using the dependencies that are in this file, you don't have to specify a conda sub. Sorry, I'll just say what conda is first, actually. So conda essentially allows you to manage dependencies. So you can specify a specific package version, and it creates a sub environment for that script, so that if there are breaking changes and you don't want to recode the whole thing, you can just call that specific package version. So by doing the way that you do this is by specifying it in the YAML file. And it's going to look something like this. So first you have channels. Just paste the rest. So for Python, you only need to put conda forge. And then for dependencies, I'm putting the packages that I need. And I guess I didn't do that, but you want to specify all of the packages that you need because this is creating a sub environment. So all of the packages that you're loading need to be defined in the dependencies. And then let's say I wanted a specific version of this. I would do OpenEO equals 1.0.0 or whatever. But right now I'm just going to use the most recent package version. And then, as I showed you briefly earlier, I can go to single script run. I can choose my file. Well, actually, sorry. First, you need to connect to the server. So you're going to go to open a terminal, make sure that it is a bash or a Linux script, it can't be PowerShell, which is slightly annoying. And you're going to go server up.sh. So I'm not going to connect to it now because I'm already connected. But if you execute that, it's going to connect you to the server. And then you can go to your browser, any browser that you want, and type localhost. You're going to see all of these different tabs. I'm going to go to single script run, where I can see if I've saved it. I can see the script that I just put in. So there it is, workshop example summarize phenology. I have the author here, the description, a link to the code, which won't work until it's merged into the main repository, but it's there. The license, the input form. So all of the inputs that I specified are now in this form. Here's what the options looks like and the multi-select options. And then you can put in all of the parameters and run script, and that's how you can test your script and see if it works. And so as you update code in this, if you're trying to debug it, as you update it, it'll automatically update it here if you are connected to the server. So that's the way that you can sort of interface with this and work with it until you get your script running. So once I've run my whole script, this is my example, I put in a bounding box of Belgium, the start and end year that I want, the season of interest that I want, bands, etc. And these are my results. So I have the outputs that I specified here. So I have the phenology rasters. Unfortunately, you can't view them here because they're multi-band, but you can view them on a different interface that doesn't work with just the script. But if it was a single band, you would be able to view it here. You can click on this and download it. This is all of the years of the raster layers that I wanted. You can look at the R code as it's executed and try to debug with that. And then this is my CSV of my summarized values. So that's just in the single script run. I've just created a script, created a description file, and then now I can run it in Bon in a Box. Are there any questions about this? I'm going to move on to quickly do an R example, but I'd love to take questions before that. All right.
Yeah, sorry, Joey, still again, I got the point here in the YAML file. So if you go back to the YAML file now, and if you're scrolling up a bit, exactly. So I mean, for instance, all the namespaces, you know, like study, underline, area, polygon, spatial underline resolution. From where do you get this? Is this predefined somewhere? So is there like a dictionary, or from why do you call it spatial underline resolution and not I don't know, something else? But from where do you get this namespaces?
So the names are just defined by the user. So when you're creating the inputs and outputs, you get to name them. They have to be snake case ID. So essentially, they can't have spaces. They have to have underscores and no capitals. But otherwise, when you're creating it, you get to choose the name. So ideally, it's something
flexible. So it's not predefined somewhere.
No. It's not predefined at all. Yeah, it just has to match up. The important thing is that it matches up between, the name is the same between your script and your YAML file so that it can recognize it. And then these labels are what's actually showing up in the UI. So that's a more like human readable thing. So yeah, like polygon of study area. instead of like polygon underscore or study area underscore. Yeah, you get.
Because they stored it
But
somewhere, maybe predefined somewhere, and you have to
no.
use this specific. No. Ah, okay, I got it. Thank you. Thank you.
Yeah, no problem. All right, so I'm just going to move on to a super quick R example, and then I'll show you how to actually build a pipeline. So again, now I've got these layers. Now I want to, this isn't really necessary to do in R, you could do it in Python, but for the purposes of demonstration, I'm going to create an R script that takes these layers and subtracts them from one another so that you can look at the difference between 2 years in the phonology, whatever phonology band or parameter you're interested in. So again, still in my workshop example folder, because it's still in the pipeline that I'm creating, I'm going to create another file. I'm going to create another file, and I'm going to call that phonology difference. I really can't think right now. .r. So I'm going to create my R file, and again, I'm going to create a YAML file to go along with that. Phonology difference.yml. Okay, so again, very similar to before. I'm going to load all of my packages. I'm going to use Terra, RJSON, which is necessary in every R script, to run in Bon in a Box, dplyr, tidyr, and ggplot. Again, I'm going to write all of the information in my YAML file. So, name of the script, the name I want to see, the author, which again is me, license, and description. So, script to calculate the difference in the layers of the phonology data. Again, very similarly, but in R syntax, I'm going to add my inputs. So I'm using this BIAB underscore inputs function so that it will read the inputs from the pipeline JSON file. And then because I want to use the rasters from the previous script that I just wrote, I'm going to make an input called input $rasters. So that's how you specify inputs in R, is input $, and then the name of the input that you want. And I'm going to choose 2 years, so a user can choose 2 years to look at the difference. So as you saw before, there were a bunch of years going from 2017 to 2024. Maybe they want to look at the difference between 2020 and 2024, but I'm going to allow the user to choose that. So I'm going to call that start year and end year. And you'll notice it's slightly different in R. If I wanted to define all my inputs up here, I could go like rasters, input rasters, start year, etc. But I'm just going to put it directly in the function. And then I'm going to go ahead and extract those with the year that I want. So I'm going to do a grepl function to find, because the file path has the year in it, I'm going to load that raster using the grepl function and my start year input. Then I'm going to go ahead and load this. Okay, so now same thing. I now have these 3 inputs. I have rasters, I have start year, and I have end year. So now I'm going to have to put that in all in my YML file. So I want this to be a GeoTIFF array because, as you saw, there were multiple years. So I want to be able to put in multiple rasters. And then the start year and end year are the same as they were in the other YAML file. So now I'm writing some code to essentially load the years that you want and subtract them from each other, and then for each band, and then output it. I'm specifying the path here. So output folder, and then the names of the files. And then I'm actually writing the raster here. So like in a normal R script, I'm actually writing the output, except that I'm writing it to the output folder, if that makes sense. And then same as before in the Python example, I'm going to output it using this BIAB output function. So the output's called phonology change. And then these are the paths of the files that I want to output. So I've created this path into the output folder with the name that I want. And then, of course, I need to put this into my YML file. I'm going to write outputs phonology change, which will show the difference in the pixel values between the start and the end year, description, and then type, which I also want to be GeoTIFF array. All right, I won't really go through this, but essentially all of this code is just creating a time series plot from the CSV file of the summarized values that I created in the Python script. So again, I have my output, I have a plot, the path that I want, so the output folder, and then the name and file format of exactly how you would output it in a normal script, except that you're writing it to the output folder. Saving it, I'm using ggSave here, and then creating my output here. I added some inputs here as well. So input time series, so that's the CSV file from before. So of course, I need to put this all in my YAML file. All right, and then again, I need to specify a conda environment. So actually, I'm going to look at the packages that I need here. I'm going to look in the runners folder for the R environment YML and see which packages are available there. If they're all available there, you don't have to do this step. But if they're not, or if you need specific package versions, you need to do this step where you add the conda environment. It's a little bit different than Python. You're going to write channels, conda forge, and then R. It's important that conda forge comes before R for whatever reason. And then I'm going to paste my dependencies here. So all of the packages that I need. Again, if I needed a specific version, I could go ggplot 1.2 or whatever. Again, if I save this, and then I go to my form, I'm going to see, oops, sorry. Yes. Not there for some reason. Let me reload. Okay, here we go. So now I have my script here with my inputs. For the sake of time, I didn't put all the inputs that are in the actual script, but okay, so now I'm going to move on to actually building the pipeline. Are there any questions about this? I know it was a lot, but there is documentation. Yes, Ben?
Okay, let's try this mic again. You can hear me?
Yes.
Can you hear me? Okay, great. So awesome. I love it. Thank you so much for all of this. I was just curious, can we install custom R libraries or Python modules?
Yeah, so you mean like in development or so
Yeah,
you
like
can
on GitHub. So I have a bunch of functions already in existing libraries, but they're not going to be, they're not on CRAN, for instance, for R.
yeah,
So, yeah.
So for the Conda sub-environment, it has to be packages that are in Anaconda, which are usually ones that are in CRAN. So you can't specify those in a sub-environment, but the way we're doing it now, and there's probably a better way to do that in the future, is you just install them in the script itself. So use DevTools, install GitHub, and then do some sort of like if else function to check if it already exists, and if it doesn't, then install it, if that makes sense. But you can use them, yeah.
Cool. And would you recommend having even like a little tool that, because you were mentioning before that it can cache the results. So if in the given pipeline, an earlier tool installs the library, that could be reused later for a subsequent, or is that an unnecessary complication?
Yeah, so originally we had it.
Detailed.
Yeah, so originally we had it where in the documentation we told people that they could add things to that like big R environment file, or if they're in DevTools, you can add it to another script to have it like essentially in the tool so it automatically downloads. But we found that it was getting a bit too bulky and was making the installation much longer. So now we're just telling people to do it in the script. But it is possible to like have it automatically download as part of the tool. It's just that we don't want like every function ever being added there or every package ever. But if it's a package that's like used really a lot, then we could put it in the environment file, if that makes sense.
Cool. Thank you.
Sure. All right. So now I'm going to show you how to actually make this into a pipeline. So I'm going to go to my pipeline editor canvas. I'm going to go ahead and clear this. Clear canvas. So all of the scripts that are available are under this sort of scripts header. And so I can see my folder here, workshop example, with my 2 scripts. But I'm not actually going to use these because I didn't fully finish them for the sake of time. So I'm going to do the exact same scripts in this phenology folder that I created before. So the first script I wrote was the summarize phenology Python script. If I click on it, I can see all of the information that I put in that YML file. I can see the inputs and outputs and descriptions as well. Oops. How do I get out of this? Not sure why it's getting so wide. Okay. So I'm going to go ahead and drag this into my pipeline editor canvas. I can see all of my inputs and outputs here as well, which is great. So if I leave these blank, they're going to render in the user form. So you don't have to do anything with these if you want the user to be able to edit them in the user form. But let's say I wanted to connect a script that takes where the user can input a country and it automatically takes the polygon of that country and the bounding box and inputs it into my summarize phonology script. And I happen to have that, and it's called get bounding box. So I'm going to drag that here. And then I'm going to go ahead and connect this polygon output to this polygon input. If you hover over them, you can see what the file format is and the description of what it is. So if I'm connecting them, they have to be in the same file format, which they are, so that's great. I'm going to connect the bounding box here. If you're paying attention, which you probably aren't, to this tab over here, as I connect things, the number of inputs is going to go down. So now this is no longer an input that the user can put in. It's an input that's automatically pulling from this script into this script. And then I also wrote the phonology difference script. So I'm going to go ahead and connect that as well. So the phonology rasters are going to go here. My table of zonal statistics is going to go here. And then the next step is to specify outputs. So there's two ways you can do this. The easy way is just double clicking. So I want my rasters to be an output as well. So I'm going to double click that. It's going to create an output. The harder way, if you really want to, is you can also drag it here and connect it. So I want my change in phonology raster layers to be an output, and then also my time series plot. And I'll add the zonal statistics as well, and the polygon of study area, because why not? Another thing you can do is, so for this script, I don't want the user to be able to change the CRS at all. So I'm just going to go ahead and double click study area EPSG. It's going to come up here. And I'm going to go ahead and make that just 4326. So now when it generates the form, the user can't change that input. It's just an explicit input. And then if you go here, you notice that there's start year, end year, and then start year, end year again. And that's because both this script and this script have start year and end year as inputs. And I don't want that, unless I wanted them to be different, which I don't. So I'm going to go ahead and connect them as the same input. So I'm going to double click on end year. I'm going to click this downwards arrow and click convert to user input. So that's going to allow the user to be able to edit it, and then I'm going to connect it to both. So you can see the number of inputs are not going down. Convert to user input and connect it. Another thing that you might want to do is if you have an input that you also want to be an output, like for example, country, if I wanted country to show in the outputs and in the results viewer, I could double click that, convert to user input, and then connect it to an output. Not many people know that, so yeah, but I'm not going to do that. All right, so this is my full pipeline here. You can click this layout button to make it sort of nicer. I'm going to go ahead and save this. I'm going to call it workshop example. This also needs to be snake case ID, so it has to be lower cases and no spaces. All right, and then when I click load from server, you're going to see my pipeline there. But I also don't want it to be called that. I want it to be called something nicer. So I'm going to go in this metadata pane, change the name to workshop example. And then obviously I should put in the rest of the information. So a description of the pipeline. So this is a description of the full pipeline. It's a bit different than just the script. The author as well, the license, any external links, and references. So if each script has references, this one's the pipeline references should be like a culmination of all of the script's references, if that makes sense, plus any other ones that are relevant. So if I save this, now you can see that it has a nicer name there. The other thing I want to show you is that these are in a weird order. I don't like that order. I want country to be first, so I can drag this and drag state and province as well. It's filling the examples from the actual script, but if you want to change the examples for the pipeline, you can do that here as well. So I want the example to be Belgium instead, a nice small country. And I want there to be no state because I want it to do the full country. I could also change any of these other inputs, and then outputs I can also reorder if I want. Make sure to save that. Now, if I get a pipeline run, my pipeline is going to show up here. So it's called a workshop example. Doesn't have any information because I didn't put it in, but you can see it auto-filled my example that I put in the pipeline editor with all of these other examples. You can see that bounding box, rasters, all of those other inputs are not showing up here because they're connected in the pipeline editor to other scripts. So it's automatically pulling those outputs from the other script into the script, so the user cannot change that, if that makes sense. And then I'll just show you what this actually looks like. So the exact same pipeline is called phenology pipeline. These are my outputs that I decided on. So zonal statistics, phenology rasters, change in phenology metrics, and plot. And that's everything that I specified with that red output box. So I can look at the results here. This looks very similar to the script output. I can look at each step of the pipeline here. So I have my summarize phenology script, my get bounding box script, and my phenology difference script. I can look at the logs here. I can look at all of the intermediate results, and I can look at the description of the script that I put in the YAML file. And then we also have this results viewer. So this is a nicer representation of the data. So here I can look at each one of the rasters. They have different bands, so you can choose the year and the band. This is amplitude, so amplitude of the phenology curve. And there's Belgium there. You can look at length in 2022. You can also look at my change for the different for the different bands. That's the change in the length of the season between my start and end year, which I believe was 2017 and 2022. Change in amplitude. And I can look at my time series plot here as well, or my table of summarized values. All right. So that completes my little demo. I know that was a lot, but again, this is all available on the documentation and also in video form on YouTube. Yeah, we'll take questions. Ben?
Yeah, thank you so much, Joy. I was just curious with, you have two spatial inputs, both the bounding box and the polygon. And I was just curious, with the difference in there, like, is it because with the raster fetching, you need a bounding box, but then and then you mask out areas outside of the polygon when you do the final calculations on generation of the rasters? I'm just kind of curious about that duplicity.
Mhm. Yeah, I'm not sure if that's strictly necessary actually, but the way that the OpenEO functions work is that when you're creating this data cube, you need a spatial extent. I don't know, and then later you can crop that by a polygon. I don't know. I think you have to specify the bounding box or else it's going to pull all of the data, which you probably don't want because it's going to take way longer, but I'm not totally sure about the like internal functioning of OpenEO because again, this is just sending it off through an API and having them process it and then sending it back. So, but yeah, that's my
So, like
understanding is that it pulls all of it and then it crops it. Yeah.
Yeah, and which is also, I'm more used to using, for instance, Copernicus marine data sets, and they similarly have that spatial extent bounding box. So as far as like tooling goes, another approach, for instance, could be, if I'm understanding correctly, is just that you have the spatial input, which could be as simple as a bounding box, and then you could have another little function in there that just gets that bounding box from the input polygon.
Yeah, that's what
that.
it's doing. That's what the script is doing. It's taking a polygon of a country and then getting the bounding box. So it's outputting the bounding box of that polygon and also the polygon itself.
Oh, but the VBox is actually an input. Isn't that correct?
So the BBox is an input of this script, but the script, it all went a bit fast. So, but the script that I connected before is a script that essentially pulls country or state or province polygons and then calculates the bounding box and then outputs the polygon and the bounding box of that polygon. So then when I connected that to the input of this, it automatically is pulling that polygon bounding box when you specify the country or the region, if that makes sense.
Oh, okay, cool.
So when I wrote Belgium, it's taking the bounding box of Belgium. Yeah. And then also the polygon of Belgium.
Yeah, I mean, I would just suggest the bounding box is a spurious intermediate result that it's a technical detail. It's unimportant here, but it reduces the clutter because the user from an input perspective and an output perspective cares generally very little about VBox. That's just an intermediate bit of machinery.
Yeah, but when you go to the actual pipeline, oops. So the pipeline that I connected here, this is the script that's getting the bounding box, so polygon. So if I could output both this and this, and then it would show them. Right now it's only showing the polygon. But no, I guess not, but you can see it here. So, I don't know why it says that, but it should be there. But there's the GeoJSON of the polygon and then the bounding box of the polygon. Anyways, we can discuss it more later, but
yeah, the message you had is probably because the new IDs might have a character difference with the previous one or something like that. Anyways, I just want to cut it short because I see people are leaving probably to other meetings, and I want to show you something before we go on break. Let me just share my screen.
Oh, sorry.
Okay. This is what I want to show you because we give a 2-week span to work on this, and there's different ways to reach to us. There is discourse. We asked all of you to create a discourse account. We can help live on the chat and spin off a Zoom meeting if necessary. And if there are questions that benefit anyone, you can create a topic, and you can also share your project. Even for the questions and the project, you could put a tag like Bon in a Box Workshop or something like this, and it's just going to group them together. But quickly, if you go on Discourse, well, there is a Start Here button, which is always a good idea to start from, which will prompt you to go to the groups on which you can choose to join the Bon in a Box group, okay? And then in the Bon in a Box group, there are a few posts like this, and if you have something that you think can be useful to others, you can put it here, some questions about Bon in a Box or contributing. There is this little DM. It's a bit subtle. This is a direct messaging, so you can click on the plus to create a conversation with Jory or myself, Jean-Michel. Hey, I can create a conversation with myself, or create a group chat if you have many other people working with you, and then you can add in the names of whoever's in your group. So I just wanted to show you this because it's probably the most efficient way to get a quick answer from us. Then if you find a bug, you can file it on GitHub on the Pipeline Engine repository. We'll share this presentation with you so you'll be able to access the links. Pipeline related issues, for example, hey, the Maxent pipeline should have this this parameter, or does not take this into account. Well, you can file issues in GitHub. By the way, so let's let's say on the Pipeline Engine repository, issues is the tab right there. And then you click new issue, and you'll be able to create a bug or a feature request. Email still works as well. And if you're unsure if you should create this here or there, well, just create it, and I can move it from one GitHub repo to another, so it's not stressful. I've put 2 links to cheat sheets for Git, because not everybody's familiar with Git as a whole. There's a lot of documentation and videos, but sometimes having a cheat sheet is quite handy. You just go here. So this is the GitHub one. This is something you can print out and leave on your desk. And this is an interactive one that I find quite cool. So if you are on the this is your workspace, so what can you do from there? Okay, you can pull, or you can clone, or so you can try to be interactive and better understand what is the workspace, what is the local depot, what is the remote. Well, sorry, mine's in French because it auto detects the language of the OS, but yours will be in your own language. So I've linked these 2 things as well in the documentation here. Before we go on break, I'll quickly mention we'll create a documentation standards document, but just be consistent in the way you name your IDs and make the descriptions to make sure someone that's outside of your organization, or maybe a biologist but not a subject matter expert, can understand what it means. Use common markdown whenever you want to, for example, you need an image in the description, you can do it. You want something italic or bold, a link, you can do all this using Markdown. So how it's visible here, for example, this is an image, or this is the italic. You can look at the hello R not pipeline script, and has a few examples of different types of output and images and italic. Other than that, the pipeline metadata filled in with clear descriptions. And once you did all this, there's a little script that runs the same thing that's validated on GitHub. So if you prefer running it locally instead of having an email telling you just pushed something that fails the tests will check for the documentation to be valid, the structure. So it won't tell you if it's good documentation, but it's going to tell you if something's wrong with the indentation or with silly things like that, or missing you created a tag for a reference but didn't put it in, or something like that. And as well, it's a good practice to put a markdown file for tutorial, or to link to an external documentation. So maybe you have a really big documentation based on your workflow, and you can link to it simply with the external link label. Otherwise, you can create a little case study markdown to walk some people through it. It's in addition to the documentation of each inputs and outputs. That can be done, of course, later on, once you're sure that your pipeline is good and that you want people to start using it. I think with this, we can go on break, and then we'll have the working period for 1 hour.